Behind the Knife, the surgery podcast. Relevant and engaging content designed to help you dominate the day. We want you, yes, you, to join the Behind the Knife team as a surgical education fellow. We're looking for enterprising surgical residents to take the bull by the horns and build something new and exciting. Fellows will benefit from ample support from the Behind the Knife team, including the use of our digital education platform and access to all of our resources like illustrators, video editing, AI tools, social media, and more. Get your name out there and build your CV by being part of the #1 surgery education platform in the world. We're offering a 2-year fellowship starting in July 2026 and ending in June 2028. Only residents beginning their 2-year academic development time will be considered. Check out the show notes for more information, including the application link. All applications are due on April 20th. Again, all applications are due by April 20th. Now, get out there and dominate the day.
Hey BTK listeners, want to feel more confident in the OR? Check out our surgical instrument flashcards. With over 300 instruments, these cards are packed with key tips, subtle distinctions, and insider tricks you usually pick up after years in the OR. Perfect for students, surgical technologists, or anyone prepping to scrub a case. You can find them in your library on the Behind the Knife app or at behindtheknife.org. Test your knowledge, share them with your learners, and as always, dominate the day.
Hi, everyone. Thanks for joining us. My name is Dr. Maddie Clunan. I'm a general surgery resident at the University of Nebraska Medical Center. And today we're talking about one of the most fascinating ideas in transplantation, and that's xenotransplantation, the possibility of using organs from other species to help save human lives. This idea has been around for decades, but today it feels closer than ever before. To understand where the field might be going, we really have to understand the story of transplantation itself. So today for the Behind the Knife transplant series, we're doing something a little bit different. I'm joined today by Dr. Joshua Mezrich. He's a professor of surgery and abdominal transplant surgeon at the University of Wisconsin. Before we get started in the actual podcast, can you tell us a little bit of why you decided to pursue a career in the field of transplantation?
Absolutely. Well, first of all, it's just wonderful to be on this podcast. I feel really honored. In a lot of ways, I stumbled into the field of transplant. I was sort of an unlikely person to even go into surgery. I kind of was a messy kid, sort of disorganized. And I wanted to be Hawkeye Pierce. So that was the one connect I had to surgery. But I went into medical school not really knowing much about surgery. And this is almost too good to be true. It sounds cliched, but it was my first day on my surgery rotation, and I found out I was on call. I was running around like crazy. I was too nervous to eat or drink. Felt like I was doing everything wrong. And I was in New York City. It was the winter. And I remember in the evening, I was scrubbed on a bowel obstruction, and I was watching these bowels move around, and I was starting to feel sick myself. And I actually remember I was hanging off of the bowels, and the surgeon was like, "Can you stop doing that?" And I just, was sweating. And somehow I got through that case. And around midnight, my resident said, "Hey, go scrub in the case next door." And all I wanted to do was go to bed. I was so tired. I felt like really out of place in the operating room. I was already thinking this is not for me. But I went in the room, and it was a kidney transplant, and the classical music was playing. It was very, very calm and quiet. I scrubbed in, and they already had the iliac vessels dissected out. I didn't know what I was looking at. And I'll never forget, they opened this cooler and they took out what to me looked like a lump of fat. I was thinking, when are they going to get the kidney? And they sewed it in, and it just seemed very beautiful and simple. And when they released the clamps, it turned pink. And a couple of moments later, urine started squirting out. And I thought, oh my gosh, I can't believe this is real. Like, how could this work? And I remember having a lot of thoughts, one, just being amazed. How does this work? Who figured this out? How could anyone think this would work? I really wondered who was that donor. I knew it was a deceased donor. And I wondered, like, who gave this gift? And maybe like the third thing I really wondered was, like, is this something I could ever do? even though I think I was unlikely to be a surgeon, I always liked the idea that if I work really hard at something, could I possibly master it? That was very exciting to me. And really from that night on, not only did I decide I wanted to be a transplant surgeon, but I decided I wanted to understand this whole field and who the people were that made it happen. So it was an epiphany. It was an amazing night in my life.
Yeah, it's so cool how, small moments can really change your whole career and your whole life. And that's a really neat story. Transplant feels like it's this established field in medicine. And I think we often take for granted just how young the field is. Can you walk us through how transplant evolved from science fiction in the 1950s to standard therapy today?
Absolutely. I think this story is incredible. And I think people sometimes don't remember or forget how new a field transplant is. Because as you described in the 50s, it really was science fiction. In the 60s and 70s, it was this highly experimental field with really awful outcomes, with a few crazy fools that were conducting transplants. And then in 1983, with the approval of cyclosporin, it became a legitimate, repeatable, non-experimental field. And one of the reasons I wanted to write my first book is that I knew that a lot of those pioneers were still alive. And how rare does that feel to be in a field where the people who actually made that happen were still alive? And I just remind people that in the 70s, which, I mean, I was alive in the 70s. A lot of people remember the 70s. Transplant was really, really tough. I mean, 50% 1 year survival, maybe a little better than that. Patients doing terribly, horrible infections, and people were just gutting it out. And there were a number of pioneers, the most well-known, Joe Murray, Tom Starzl, Roy Kalm, a number of other people, all of whom have died in the last few years, who just had the courage to make this field happen. They just knew it was going to work out. And I wanted to have that opportunity to talk to them and understand how they could do that. I don't think I could do that. I can do what other people teach me, but in general, if you're operating and you're doing something no one has ever described before, you should be a little cautious, be a little humble about that. But that's what these people did.
Yeah, it is pretty amazing that they push past, because it seems so crazy, right, to push past the outcomes that they had in the beginning. But I mean, now today, it changes people's lives every single day.
Yeah, I mean, I think when I spoke to them, I didn't want to know what they did, because I knew what they did. I'd read everything, that they'd written and that others had written. But I wanted to know, how could they persist when people were calling them murderers, were not sending them patients? There were petitions with Starzl in particular, petitions to have him fired from basically every place he ever worked. Someone wrote a book about these early surgeons, and the book was titled "The Courage to Fail." And I do agree. It was written by a sociologist who followed them around. And I agree that they had the courage to fail. But I kind of think a lot of us have that in our field and in other fields. But to me, there's something subtly different, which is that they had the courage to succeed. They knew it was going to work out. With each failure, they said, I'm going to learn from this, and this is why it didn't work, and I'm going to keep going. And when you talk to Starzl, he said, it had to succeed. I mean, it just, my entire life would be a farce if it didn't. I just knew it was going to succeed. And, each one of them was different, but they all had this belief that no matter what, it was worth persisting.
I love the history of transplant. And I think xenotransplant is a really nice, it seems crazy, and it's always seemed crazy, but it's kind of gone along with the whole history of transplant. So I know in your new book, you describe xenotransplant as kind of unfolding in several eras. So should we start with the primate era?
I think that makes sense. In a lot of ways, the idea that you could take an organ from someone who had died, and back then they didn't have preservation solution. They didn't have brain death. That wasn't defined until 1968. So these were true cadaver organs. I mean, the thought that would work seemed totally crazy. And actually in those early days, because they living donor was risky. They didn't really have immunosuppression, so primarily they were doing identical twins. But the idea of using a primate as a donor, was not hard to get your head around. I mean, primates look like humans. They share a lot of genetic similarities, particularly chimpanzees. So really, since the earliest days of transplant, people were thinking about xenotransplant. And, maybe the maybe the father or the patriarch of xenotransplantation would have to be Keith Reemsma,
Okay.
who was a larger than life figure. He was chair of surgery at Columbia in New York for many, many years, but he actually, his parents were missionaries and he grew up out in the Southwest. He always wore a cowboy hat and cowboy boots.
Oh,
Cowboy.
really?
He was a larger than life figure. He was in the Korean War, actually shared a tent with Frank Spencer, who was a very famous heart surgeon in New York.
Okay.
The two of them were residents, like second and third year residents. They operated like crazy. They flouted the rules. The military had these rules that when there was a vascular injury in the leg, they had to do an amputation because
Oh, interesting.
that was from World War II because they didn't have helicopters, and by the time patients got brought out, their legs were dead, and so they still was in the rule book. In Korea they had to do amputations, and Reemsma and Spencer were like, we're not following these rules. So they were just doing bypasses, almost got court-martialed. And I think Reemsma developed this attitude of like, I don't care about the rules.
Mhm.
I'd rather ask for forgiveness than permission. He was, as an aside, many people think Hawkeye Pierce was based on Keith Reemsma. He showed up.
Oh, really?
He showed up with a crate of scotch and apparently was always mixing martinis. And Alan Alda went and visited him when he was first filming as Hawkeye Pierce. So part of the legend. But when he got down to Louisiana, he wanted to start a heart surgery program. He was down at Tulane, but there were no patients because, well, they weren't doing bypasses. And he wanted to do something new and impactful. And he wanted to do kidney transplant, but there were like no donors other than identical twins. So he got the idea, if a patient has no donor and they couldn't do long-term dialysis, I'm going to offer them a primate transplant. He did one monkey and that went badly, so then he switched to chimpanzees. He transplanted 6 patients, and the only immunosuppression they had was steroids, radiation, and they had Imuran just by that point. So not very much. And many of those patients did badly, but 2 of those patients left the hospital. And one young woman not only left the hospital, but she went back as a teacher and lived for 9 months with perfectly functioning kidney. And unfortunately died from an electrolyte disturbance at 9 months. So to this day, that's the longest surviving xenotransplant. But I hope that record gets broken soon.
What do you think the early primate era taught the field of transplant?
Yeah, I mean, I think it taught them a number of things. Number one, it taught them that it probably could work. If immunosuppression got better, it probably could work. It taught them that genetic matching, which was very much in its infancy, probably mattered a lot because Starzl got involved in this too, and he analyzed baboon results and chimpanzee results, and the chimpanzee results were so much better, which he ascribed to the genetic matching. It also taught them importance of public trust and the risk of pushing the ethics of using animal organs for humans. And, Reemsma, in my opinion, was a very ethical man. He was very open about what he was doing. He was very clear with his patients about the risk and really not knowing if this was going to work out. And he strongly felt it was okay to use chimpanzee kidneys. I don't think the public really had a huge problem with it because I don't think they associated kidney from a chimpanzee that it would necessarily kill the chimpanzee.
Oh, sure.
I mean, of course it would. We know that, but I don't think that the public knew that. And it wasn't until the Baby Faye case in 1984, which was the baboon heart, where the public exploded with protesting, the idea of using a primate organ for a human.
Can we talk a little bit more about that story, the Baby Faye story?
Yeah, I think Baby Faye is still the most famous xenotransplant case around. And certainly, I think anyone who was alive and of age in 84 might remember that case or something about that case. It's really interesting. The surgeon was a man named Len Bailey who had trained in Toronto. He was an excellent heart surgeon, and he wanted to do pediatric heart surgery, and he really believed in transplant. He thought the reconstructive surgeries on pediatric hearts were terrible. And primarily, he was talking about hypoplastic left hearts, and those kids would just die. And it was just at the very beginning where surgical options were coming around, the staged procedures for hypoplastic left heart, but they hadn't even done like the second stage yet. So pretty much it was a death sentence. And Len Bailey had this idea that since infants have, lack a really well-developed immune system, that, maybe a heart transplant could work for those kids. He actually did research in the lab with goats and sheep, and he wasn't having good outcomes. But then when cyclosporin got approved in 83, he got hold of some, and he had good results, like between goats and sheep with cyclosporin, with baby goats and sheep. He thought that was a big difference. He got referred this patient whose name was baby Stephanie, and she was, blue, she was dying, and it was past the point where they could even possibly offer some sort of stage reconstruction because she was just too sick for that. And he had a long discussions, 7-hour discussion with, I guess, Stephanie's mom. And he was very honest about everything, and they ultimately decided to move forward with a baboon heart transplant. There's some challenges there. I mean, no one was doing infant heart transplants in any way, and he wanted to do a human one, but it was like literally impossible to get a baby heart. Like he couldn't. So it basically was do nothing and let Stephanie die or do the transplant. They decided to use a moniker, a fake name, because they knew that a lot of press about this. So they went with her middle name, which was Faye.
I see. Okay.
And they did the cross matching that they could. Interestingly, baboons are mostly blood type AB. Faye was not compatible with that, but he thought because Faye was a baby, she probably wouldn't have significant antibody. Chimpanzees, there are blood type O and A chimps, but there are very few baboons, interestingly. So we went ahead with that transplant, and actually the surgery went like amazingly well. And baby Faye initially did amazing. There were all these amazing pictures of her, like, just around the nursery and with this big phone next to her because her mom was sick. And so they kept her outside. But the public went crazy, massive protests, threats. Len Bailey had to wear bulletproof vests. He was always with police protection. The police would open his mail. His kids couldn't go outside. People were calling him, a murderer for baboon donor who happened to be named Goobers. Probably not ideal to name the donors. That's something I think people learned. But the baby was doing great. And then unfortunately, after about 3 weeks, she ultimately got sick and did die. But Len Bailey wrote some very beautiful things, very beautiful papers and press releases, discussing and thinking about this idea of using animal organs for humans. He said he did struggle with it, but he kind of thought, the lion eats the zebra, there's a natural order of things. And if he can save a human with an animal, he thought that was appropriate. Other people may disagree with that. There's some people who use this term speciesist, which is similar to I guess racism, but between species. That was not really a term that was used then, but became popularized later. Ultimately, after Faye died, Len Bailey had another baby that he was considering doing this on, but he didn't want to put his own family through everything they went through. But amazingly, he got a call. He had a baby who was dying of hypoplastic left heart, and this surgeon from elsewhere said, "Hey, I know what you did with Faye. Believe it or not, we have this baby with, I think it was hypoxia or anencephaly or something." And he was able to get that heart and perform the first infant heart transplant. And that baby, who they nicknamed Baby Moses, because they knew, is still alive today.
Oh, very cool.
Yeah.
You really?
And he basically created the field of pediatric heart transplant. He did 350 infants in his career with amazing outcomes. So he sometimes gets vilified, but I believe he was courageous. He gets accused of not having done the work and have had the data, but actually he had quite a lot of data, but he didn't have a lot of publications. And he thought cyclosporin was going to be a game changer for infants, which is not the craziest idea. I think it's likely that in this era, if one were able to use chimpanzee organs with the immunosuppression we have right now, we probably could make it work. However, the ethics of trying to breed chimpanzees
Yeah, totally.
For humans is quite dystopian.
Yeah.
And then I have to, so the person that maybe put the fork in the primate era, believe it or not, I think this is in the book. I should do it as a teaser, but I just got to tell the story. Of course, Jane Goodall, who
Mm-hmm.
just died recently, the primatologist, the absolutely brilliant, fascinating trailblazer, who studied chimpanzees and other primates over her career, was probably the person who ended the primate era. Was it the 25th anniversary of Riemsma's xenotransplants? They had a conference in New York. And the organizer decided to invite Jane Goodall. He's a transplant surgeon. And I got to talk to him. And why would
Yeah.
they're like celebrating Xeno and they're all pro Xeno. Like, why would you invite Jane Goodall? This is 1989. And he was like, well, I thought she was part of the story. He just wrote a letter to her organization
Uh-huh.
and she agreed to come. And she showed up and they all presented their data. Starzl was there and Riemsma was there,
Uh-huh.
Kahn. And then they were presenting the basic science and the immunology assays. And then in the last session, they had Jane Goodall come. And she gets up and she says, you know what? I'm not going to talk to you guys about any data. I'm not going to talk to you about whether what you're doing is right or wrong. I'm just going to tell you a story about the family I've been living with for the last, 30 years. These chimpanzees had names, they had emotions, they had love and hate and envy, and they used tools and, everything you would equate with a conscious being. I mean, you have to remember back in this period, people would argue, are these animals like conscious? It's sort of crazy
Yeah,
now,
it is
right?
crazy.
At the end of this talk, basically everyone was like, we can't do this anymore.
Mhm.
And that was the end of chimpanzee transplantation.
That's fascinating. I love that story.
Afterwards, she was in an interview and she goes, I just thought those people were so crazy.
So as after the primate era ended, can you take us through the next era?
Yeah. So interestingly, even before the primate era was over, there were a number of very brilliant researchers who knew that primates weren't ultimately going to work out. And they knew that for a number of reasons. Number one, well, size. I mean, chimpanzees get up to like 100 pounds, but the other, monkeys, baboons, they're really small. And they would transplant both kidneys en bloc, kind of similar to how we do a peds en bloc transplant now. But they still were way too small, a 30-pound baboon or whatever. And so that was number one problem. Number two problem was they breed like we breed. And so not only would it be, as I said, kind of dystopian to have like an island of breeding, chimpanzees don't, breed well in captivity. But then even beyond that, they already were understanding that ultimately they would need to do gene modification and gene editing. And they kind of were aware that without a faster breeding cycle, that was going to be impossible. And then infectious risk. I mean, HIV came from chimpanzees. Some of the hepatitis are in chimpanzees and a lot of other diseases. So that was a concern. Yeah. And then the public response to it. So pigs became a natural choice for a number of reasons. Number one, we sacrifice so many pigs for food that nobody, a billion a year, nobody seems to mind that. So it's kind of hard to not arguing against that. Number two, they breed very quickly. They have big litters. You can have a litter up to 8 pigs. They can breed again and again. So that works for gene therapy. If, they didn't know how to do it then, but they knew eventually that would work. And we'd already been exposed to pigs for so many years with our interaction with food that the thought was that the infectious risk would be lower. So there were a number of researchers, and they found out very quickly that if you just put a pig organ into a human or into a primate as a model for a human, into a baboon or a monkey, they would rapidly reject the organ. And people like David Sachs, David Cooper, David White, weirdly, like there are like 5 Davids as it's apparently you have to be named David to be a pioneer
Have
for xenotransplant. But they ultimately were able to identify that humans and primates, old world monkeys, have natural antibodies against any animal below that on the evolutionary scale.
Okay.
And they kind of sorted out that that antibody actually evolutionarily probably was very important. Probably a virus or bacteria came around where those primates, like 28 million years ago, that had alpha-gal antibody survived this epidemic or pandemic. And then ultimately we evolved from old world monkeys. Interestingly, new world monkeys, which had already migrated elsewhere, don't have alpha-gal antibody, which is thought to be that they weren't exposed to this. So it really, really fascinating.
Interesting, yeah.
So then they spent like the next many years trying to figure out how can you make a transplant work when you have tons of natural antibody? Is there a way to get rid of it? And ultimately, it became obvious that they were going to have to find a way to get rid of this sugar molecule from these pigs, and that was going to require gene therapy.
Mm-hmm.
So that, yeah.
The late 1990s and early 2000s were also the era of cloning. How did these advances influence xenotransplant?
Yeah, I mean, maybe we'll talk about this more, but I think like most scientific advances, there's incremental hard work. And I don't say that in a belittling way. The incremental work is critical and it's fascinating and it takes hard work. And then there are these episodes of quantum improvement. So like, I don't know, I think in human transplant, maybe the quantum was the identical twin transplant because it proved to people in 54, because that proved to people it could happen. Then the incremental work of trying to make it work. And then cyclosporin was like the quantum leap in 83 that made human allo transplant work. I think for xeno and many other areas of science, being able to clone Dolly the sheep was a massive quantum jump, because before cloning, they had these very primitive ways of doing gene therapy where you literally would take some DNA and you would inject it into like a fertilized egg. And this worked well with mice. And actually, David White was able to do it with a pig. He made a transgenic pig, but you like inject DNA into tons of fertilized eggs, just hoping it would get somewhere that it would get made and not disrupt something else. And then, just put this fertilized egg into a sow. And then you do this for years and maybe you get one pig that had one gene, but you couldn't do a knockout like that. There was no technology to do it. And people understood that you would need to be able to do your gene editing in cells in a dish that were dividing. And then you would need to be able to take those cells and clone an animal. Without being able to clone, you couldn't do anything in a dish with dividing cells. It was just impossible. And people thought they were able to clone mice actually back in the 80s because they found stem cells that would divide that were really easy. But in large animals, they couldn't. And then there were a number of different groups. But the group in Edinburgh, they figured out how to clone Dolly the sheep. They took mammary cells and they got them to divide in culture. And basically, they sucked out the nucleus of the mammary cell and they put that into an egg cell that didn't have a nucleus. So that's like nuclear transfer. So you take this nucleus out, squirt it into the egg, and then put it into a sow, and they were able to clone Dolly the sheep. And then shortly thereafter, they made Polly the sheep, where they actually took that mammary cell and they put in a gene for enzyme in the milk that could cure human disease. And that's where their idea of like, oh my gosh, we can use cloning to generate a transgenic animal that can make something that can cure disease. The group that did this, they were scientists, but they had a funding arm that recognized that cloning was going to be huge. And they also recognized that it might lead to xeno, and which they knew was going to be a big market. Novartis was already pretty interested in xeno at this point. So they started a company called PPL Therapeutics that ultimately down the road was bought eventually by United Therapeutics, which is one of their companies in xeno. So like there's a great story of a guy, another David Ayers, who's now the head of one of these companies. And he was a gene editor. He was working in mice, and he was looking for a new job, and he got invited to PPL in Edinburgh, and they wanted to open a branch in Virginia. And he was like, this was before Dolly was cloned. And he was like, okay, yeah, maybe this will be interesting. And he was trying to decide whether to take the job, and he was on vacation back in the States, and he was reading Jurassic Park, and he was like laughing to himself, like, this book is so stupid, this will be done. Halfway through the book, the New York Times article came out about cloning of Dolly the sheep, and, the world's going to change. And he said he just like closed the book and threw it out because he was like, it's so banal. It's not even worth reading anymore. And that's how big this was. I mean, people knew immediately that the world was going to change. Dolly was cloned because they knew even with at this point, they could do homologous recombination. They could do some very primitive gene editing. And they knew once they could clone, they could make knockout animals. It was going to be painstaking, but they knew it could happen.
You talk about quantum leaps in the field of transplantation. What is the next big thing that changed xenotransplantation?
Yeah. So there were a couple of there was the good and there was the bombshell. I think the good is that two groups, one David Sacks's group ultimately at Mass General, and I worked in his lab, but not a little bit before that, as well as the David Ayers group working for, that PPI, which ultimately became United Therapeutics, were able to make an alpha-gal knockout pig. And they made that kind of 2002 into 2003. And they did it using homologous recombination, so early version of gene editing and cloning. And it was like a huge effort, and it seemed like it was going to be a world changer. But as they were doing this, there were problems arising. And maybe the biggest problem, two problems. Number one, animal protests. So the UK has a very strong history of animal rights groups for hundreds of years, much more so than the US. On top of that, they actually went through the whole mad cow sort of epidemic, I guess you'd call it, where, people were getting really, really sick from cow meat. And so this combination of thinking that it was a bad idea to use animals for humans, both because it was unethical, but also because it was risky, actually kind of ended Xeno in the UK. So David White, who was working in the UK, got kind of run out of town, and Xeno moved over to the US. The other kind of bad thing that happened to Xeno is this endogenous retrovirus, sadly called PERV, porcine endogenous retrovirus. It's identified. So people might not realize that we all have viruses that are embedded into our genome, which some people call endogenous fossil retrovirus, and they actually are important in evolution. So the fact that humans can make a placenta, or women can make a placenta, came from an endogenous virus. So those viruses, some people say 8% of our genome comes from viruses, and they get passed down from generation to generation. But these viruses lay dormant, but they can wake up. Rip Van Winkle, that was written by Spike. And so a researcher, a very high-end researcher who was doing a lot of HIV research, published a paper showing that pig cells can have this virus, and in culture, the cells can infect other cells. So that was a big headwind that, wait a minute, if we actually put pig organs into humans and immunosuppress them, it could unleash a pandemic. Nevertheless, these two Davids pushed on and, got to the point where they had these alpha-gal knockouts, and they, at the time, were being funded by big companies like Novartis, and actually Starzl in Pittsburgh were funding the other group. And they got to the point where they were able to transplant these pig organs into primates. And while the outcomes were dramatically better than ever seen before, they were able to get a heart sewn into the belly of a primate to last, almost 200 days, and they got a kidney to last close to 100 days. It wasn't so dramatic that it was going to enter the clinic anytime soon. And so this is around 2003, 2004. The big companies like Novartis said, between animal rights, this infectious risk of PERV, and the outcomes aren't moving quick enough, we're pulling all our money. Xeno went into the wilderness. I mean, with still incremental work funded by NIH, funded by other government, agencies. UK still funded some research, although not at the level it used to. Xeno kind of went into the wilderness and was, had the risk of not advancing. And, I would say people made incremental progress, but probably the next quantum leap would have to be CRISPR-Cas9. Because at this point they had better gene editing techniques. They had this thing called zinc finger endonuclease, where they could actually do gene editing a lot better than homologous recombination. They could have a guide and they could cut genes out, but it was still highly inaccurate. Took a lot of effort. They didn't always know if it would hit the right spot. So they were still doing gene editing and cloning. They actually made more knockout pigs, but nothing was kind of really exciting industry and really generating a lot of interest. But then a combination of the Human Genome Project, understanding that we were getting our heads around the genome, and then this idea of CRISPR-Cas9, where people realize we can really easily edit genes. We can do it at will. We can do it accurately. We can do it quickly. And then with CRISPR-Cas9, all these big companies, all these big investors were looking for people and companies to invest in, and Xeno became one of those things. I don't think it's fair to give all the credit to CRISPR-Cas9 because I think some of the really good gene editors of that era will say, oh, we were doing pretty good gene editing already. That was a game changer. And I mean, a bunch of really big names got into the game. So, like George Church, who many people will have heard of, the Broad Institute, a Harvard researcher who I describe. He's this tall, lanky guy. He has a deep voice, and the person he most resembles is God. Like he looks like my image of God. But he's done a lot of amazing things. He's the guy who's trying to bring the woolly mammoth back.
Okay.
My brother actually wrote a book about him and that.
Really?
He spun off like tons of big companies, but he got interested. He was a gene editor, not a transplanter, but he was like, what cool things can I do with CRISPR? And he cut out the entire PERV virus, which was like 59 edits, and added 10 additional edits that were both knocking out alpha-gal and a few other things and then adding things in. So he has a pig that has 69 gene edits. It's the most edited transplant on Earth. Some other fascinating figures got in the game and who I write about. So it's funny. The book, like the first half, goes through the primate era, the history, the incremental work, the Davids, gets us to that point. The second half is almost like a movie where it's like, give me like some amazing characters that are going to make this happen. So you got George Church, you got Martine Rothblatt, who, in my opinion, could be the most interesting human being in the world. And many listeners will know Bob Montgomery, kind of ultimate pioneer, also on the short list of most fascinating people in the world. So Bob is a transplant surgeon, was really big in desensitization and paired exchange. He himself has a congenital heart malady. He actually had 7 cardiac arrests, including at a Broadway show where they did CPR for like an hour. And then he woke up and looked around and waved his hand and the whole theater erupted. He arrested in Patagonia on a hunting trip. And somehow he lived through all of this and ultimately got a heart transplant from a Hep C donor. He was one of the first. And when he was lying in the ICU, he thought he might die and he felt bad taking a heart from someone else. And he said, if I get through this, we need to find a new paradigm. We can no longer live under this paradigm where someone has to die for someone else to live. We have to do something different. It's gone all in on Xeno as well. So I think the quantum leap was CRISPR was this ability to make really complex, amazing pigs. Now there are multiple pigs out there with anywhere from 3 to 10 to 69 gene edits. And in primates, we're able to have those organs, whether they be hearts or kidneys, last multiple years. And this work has now led to humans getting transplants. And now our first ever human trials, we have 2 human trials that are happening now.
Can you talk a little bit more about where Xeno is today?
So I get goosebumps thinking about this, that we finally reached this point. I have to say, when I was in the lab, in 1999, I thought this would actually come quicker, but it's just a foolish. And I kind of thought the first alpha-gal would be game changer. But we finally got to a point where the pig to primate data was good enough, with primates lasting more than a year, that it became time to try on humans. And the FDA has, although I think they've been overall supportive, challenging in many ways too. So they did approve a number of transplants through compassionate use. And I think there were 2 heart transplants performed at University of Maryland with Bart Griffith and Mohammed Mohudeen, who's another fascinating character. They were able to do 2 humans who were turned down for various reasons for human heart transplant, were really, really sick. And some ethical challenges there, whether that's appropriate in people that were turned down for human transplants. And they received hearts. Their hearts lasted a couple of months. They worked really well initially. They got off the breathing tube off. These were really, really sick patients, but they died of various things. And then a number of kidneys were done. I think Mass General did the first kidney, which was an eGenesis kidney, into an African-American man retransplant. His kidney worked really well for a month, and then he suddenly died of a heart arrhythmia, may or may not have been related to the transplant. And then Bob performed a kidney into a patient that got a VAD. You can't get a VAD in renal failure, so it was the only one. And it was very fascinating. She did really well initially and also unfortunately died. Then they did a second kidney into a woman who actually had been a kidney donor
Really?
down in Alabama and then was very sensitized. And she got a kidney and did really well, got out of the hospital, but unfortunately lost it after a couple of months. And then Mass General did a guy from New Hampshire, and he did amazing for just under 9 months. Perfect kidney function, out of the hospital. And CNN did a piece on him with Sanjay Gupta. They were taking a walk together. Really good. But unfortunately, he lost his kidney, and he lost it like 1 week before he beat the record from Keith Reeves. So sort of ironic. But anyways, 2 trials have been approved so far. One with United Therapeutics, which is Martine Rothblatt's company, and Bob Montgomery is one of the surgeons doing the transplant. He's the chair at NYU. And they haven't announced how many they've done, probably a couple. The trial is they have to do at a couple centers 6 transplants, and as long as they have, then they're going to see how they do for a few months, and as long as they do well for something like 6 months, it's going to go to a multi-center 50-person trial. And then eGenesis also has started a trial with Mass General being the lead center, and they've also done some, and it's a similar trial. I think they have to do 3 more transplants, and then they'll go to a 20-person multi-center. So hopefully in this calendar year, hopefully we'll get into the multi-center trial. I think that right at this moment and there are a few different pigs being used, the 69 gene edit without perv, the 10 gene edit that United Therapeutics uses, which has knockouts and some knock-ins of immunomodulatory genes. I think realistically, we can get 6 months to a year survival of these kidneys, and they can function well as a bridge. I am unsure that we can get longer than that, both between the challenges of how much immunosuppression they need and the potential for increased risk of rejection. That said, there are some newer pigs out there. Joe Tector has a newer pig that's kind of hot off the press, so probably not yet ready to, he probably wouldn't want me to talk about it too much, but seeing some amazing results. And so my gut feeling prediction here could be wrong, is that I could see this iteration of pigs, and there's a few other things going on. I couldn't get into everything, right? This version maybe could see FDA approval as a bridge. What are the scenarios? Someone who couldn't tolerate dialysis, maybe someone who's highly sensitized, maybe pediatric heart transplant, because they don't have VADs or mechanical hearts for peds patients. So that could be an area. I think next iteration, in the next 5 to 10, hopefully less than that, is going to be significantly better. I hope a decade from now, we've got xenotransplants that can be similar to humans. I hope in our lifetime, or maybe your lifetime, because I'm aging rapidly in this career, but, we can have pig organs that require less immunosuppression, maybe even tolerance, that can last longer than humans, that can be ordered, that can even be matching genomes, or at least matching class 2. I think all of that is possible because our ability to gene edit and cloning has just gotten so much better so quickly.
It's such an exciting time to be in transplant. I mean, it's just, it still feels a little bit like science fiction, but it is happening today.
It's happening today. There's so much. I just want your listeners to know, this is one area. I'm a xeno optimist. That's my point. And I love xeno. I think it's I think you can't think of transplant as what it is now. If we got to a point where we had unlimited organs, it would be such a different field. Now we only list people that can get really good outcomes, but there are, 5-6 million people that have heart failure in the United States. There countless people with cirrhosis, with lung failure that we don't even consider for transplant. So I think the sky's the limit. But there's so much buzz going on in the field of transplant. This is just one of many innovations. I think transplant's a really new field, but there's so many exciting things going on that it's sort of electric, actually, to practice in this field right now.
Will you tell listeners a little bit about your upcoming book and where they can find it?
Absolutely. So, the book is slated to come out April 7th. People may not know books always come out on Tuesdays. That's sort of a relic of of times when people would go to the bookstore and buy books. So April 7th is the release date, and you can pre-order it now either through MIT Press or Penguin Random House or Amazon, always the easiest one, or from your local bookstore. And then you can actually buy the hardcover on April 7th.
This has been a really interesting conversation, and I'm so excited for all the listeners to check out your book.
Yeah, I mean, just thank you so much for having me on here. And there are a million more stories, a million more people that are part of this. And I think the future is going to be really exciting in our field. Despite all the crazy things going on in the world, I still remain excited about transplant. Dominate the day.
Be sure to check out our website at www.behindtheknife.org. Download our free app available for Apple, iOS, and Android. Simply search for Behind the Knife in the App Store or Google Play to download the app. In the app, you can listen to our episodes, watch high-yield videos, and even access our premium content, including our oral board reviews and trauma video atlas. You can also follow us on Twitter at Behind the Knife and on Instagram at Behind the Knife Podcast. If you like what you hear, please be sure to leave us a review. Content produced by Behind the Knife is intended for health professionals and is for educational purposes only. We do not diagnose, treat, or offer patient-specific advice. Thank you for listening. We appreciate your support.
Until next time, dominate the day.